You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with Future Tech and Future Tech Health Podcasts, and I have Kamal Obbad. He's the CEO and co-founder of Nebula, and the website is nebula.org. So, Kamal, thanks for coming. How are you doing today?
I'm doing great. Thanks for having me.
Yeah, tell me about Nebula. The only nebula I know of is like in outer space, a bunch of stars, but what's the premise of the company?
Yeah. So Nebula is, our mission is to make genomic data as useful as possible for medical researchers. So we were founded about a year and a half ago, and we spun out of George Church's lab at Harvard Medical School. If you know who George Church was, I think you guys may have had him on the podcast before.
Yeah, I spoke to him. Good guy, big beard, knows a ton of stuff. So, yeah.
Looks a little bit like Santa Claus. Yeah, exactly. George is, one of the pioneers essentially in genetics, right? So he's been involved with genetic sequencing technology since the beginning, from the Human Genome Project, all the way to Nebula today. And there's been this focus for the past 15, 20 years on let's bring down the cost of whole genome sequencing. And once the cost gets low enough, from billions of dollars for one genome to $1,000 today, we're going to see this, we're going to reach this inflection point where a lot of people get sequenced, a lot of data is generated, data becomes accessible, researchers can do cool things like rational drug design, like doctors can do interesting things like data-driven clinical decisions or personalized medicine. And what we've seen, in recent years, we hit that $1,000 price point. The price has gone down a ton. We haven't seen a similar increase in adoption. So Nebula was created with the idea that, there's a lot of other issues in genomics today that are preventing it from, doing the things or being utilized in the ways that it can be by researchers. A lot of data is fragmented. There's huge privacy concerns. A lot of genetic data that's being created today isn't whole genome sequences, right? It's SNP chip or genotyping data. A lot of the data is being generated today.
Tell me about that for a second, because I don't think most people would understand. So, when you say sequence the genome, I would just assume you'd sequence my entire genome, but services like 23andMe and places like that, they're not doing all that. What are they doing in comparison?
Yeah, so there's different ways to essentially read a genome. Most people, when you're doing the direct-to-consumer genetic test, it's usually what we call SNP chip or a genotyping array. And what this means is you effectively are looking at a very small fraction of your DNA. So usually, a fraction of 1% of your DNA, and you're looking at really the subset of your DNA that we know what it does, right? So if you have an A here instead of like a C or something, we know that variant is meaningful. It has some potentially clinical or trait significance. So that's typically what's being done when you do genetic testing, and that's fine, right? Because it's pretty accurate. It usually is you're testing the most important parts of your genome that we know about today. But it's not that great from a researcher's perspective, right? Because we're not generating whole
because it's the known part, and you said it's like 1% of all the genome. So as the unknown shrinks, as we figure it out, why not have the whole genome so that you can use that data to figure out the rest of it? Otherwise, it's like a catch-22, right?
Yeah, exactly. And it makes sense for a direct-to-consumer product, right? People buy these sequencing genetic tests essentially because they want to get a report. So why should we test the parts of your genome that we can't give you a report about, right? Because we don't know what they do. Which is bad for researchers, though, right? Because researchers want those parts of your genome. They want to study that part. They want to understand how it influences your health, your traits. And most of your genome we don't understand today, right? And that's because there aren't a lot of whole genome sequences that exist. A lot of people have done SNP. I think we have tens of millions of people in the U.S. alone who've done like 23andMe or Ancestry. But that data isn't providing enough value today. So,
well, what would be a good offer is to go to those people that already have done that part and say, hey, once it gets cheap enough, hey, come back and do the whole thing, because those people have already been willing to do it and aren't scared of it, and maybe got some knowledge from it and they'd want to get more. Yeah. So that might be a great population to market to.
Yeah, that's a great point. And I know a lot of these direct-to-consumer companies are biobanking samples, right? So they collect your saliva and they keep it in a fridge somewhere, because that is the goal, right? When sequencing prices get cheap enough, they will hopefully do whole genome sequencing. But it's still tough because the whole genome isn't useful on its own. You really need it attached to the person, right? You need to understand the person's health. You need to understand the person's medical history, their family's medical history. Once you get a very holistic and longitudinal view of that person's health over time, then you can start understanding how the genome influences that. So that's really the big problem right now in genetics. It's very hard to get sizable genomic data sets that's also paired with rich phenotypic and clinical data that actually makes those genomic data sets valuable for researchers.
So how big are the data pools out there? Does does one institution have 1,000 people's sequence and phenotype data, and then the other one has 100? You have to piece them together, or what's out there right now?
Yeah, it's a good question. There's probably, I think there's a couple million people, last time I checked, who've done whole genome sequencing, and the data exists in various places. So there's some large data sets. There's Genomics England, there's Genomics Ireland. They have on the order, I think, of hundreds of thousands of people who've done whole genome sequencing, right? And they have their medical histories and all that. And this is essentially government-funded projects. You have UK Biobank. It's not whole genome sequencing, but it's similar. It's exome sequencing. They have 500,000 people. So there are data sets being generated. You had Decode around 10 years ago, right? I think they had around 1,500 or 2,500 whole genome sequences with rich clinical data that they paired it with. So there are data sets out there, but even hundreds of thousands, you get into this issue of dimensionality, right? A genome is billions of base pairs. Hundreds of thousands of genomes, that still isn't enough, especially when you're looking at rare conditions. Let's say you want to understand a rare form of cancer or a rare disease. You're not going to find hundreds of thousands of genomes that exist for that disease. You'll go to the UK Biobank, you'll go to Genomics England, and you'll maybe find a dozen, right? So we've talked to a lot of these pharma companies, and it's a big problem they have. When they're talking about doing genetic studies for rare diseases or genetic conditions, we're not at the point today when we're talking about, let's get 100,000 genomes of people with, this tumor that responded to this drug or whatever. We're really just talking about
why do that? Why not focus on the common variations instead? Because you're much more likely to sooner get that information, and that would tell you about the mechanisms of genetic change, whatever they may be, or effect, and then go on to rarer stuff later.
Yeah, I think a lot of interest right now is in, when I say rare diseases, you obviously can't see me because it's a podcast, but I'm doing air quotes, because they're rare diseases, but they're still affecting, millions of people, right? So they're diseases that have, we think have high genetic components to them that we don't fully understand. And it's hard to get. Those are diseases where the genetic data is very valuable. It's very hard to get enough of it. And there's just so many barriers that make it hard to get enough genetic data. Some of it's regulatory, some of it's even cultural, right? People are scared of getting their genome sequenced. And this has been a recurring problem in industry for researchers, for biopharma. It's just that the data acquisition costs are so high. That's become a barrier to actually doing large-scale genomic data-driven research.
Okay. Any mathematical modeling that's been done so your benchmarks? Oh, we need 3.5 million sequences in order to be able to determine X, Y, and Z. Do you know what your goals are? That's
a really good question. No, not that I know of. It's definitely possible somebody's done something like that. I'm not exactly sure what those numbers are. It's something I need to look up.
Well, the reason why I say it is like you're saying, oh, we need more genome sequence and all that, but if you don't know, like you'd want to get to a milestone as soon as you could. Let's say the number was only 220,000 for a particular set of conditions, and you want to get there first, so you can publicize that. Say, hey, we've got enough data now to look at A, B, and C. But probably some mathematical modeling would do that for you, and to look at various things.
I think you could probably do something like that for a lot of use cases, and it would work to give you a ballpark for what you need for statistical significance. But a lot of the stuff we're seeing in the space are researchers or biopharma paying a ton for genetic data, and they don't have a clear understanding of what the potential ROI is going to be. It's kind of like an analogy I use a lot. It's kind of like land speculation, right? You go out, you buy a big plot of land, and you start digging for gold or digging for oil, and you hope you find something. A lot of the genetic big data plays have kind of been like that, right? Where, you talk to people in industry and they say things like, oh, we don't know if it was worth it. We don't know what they got out of it. They spent $500 million in this large genomic data set.
Right.
Maybe they got no return on that investment, right? So a lot of it is in early days where we think researchers, there still isn't a clear sense of how to value these types of data sets. Should we just go out there and sequence a ton of people? There are some arguments, there are various arguments on this. Some researchers, some papers out there that say, at population scale, whole genome sequencing makes sense, right? Because if you whole genome sequence everyone, you're going to be saving healthcare costs in the long term, because there's enough people with preventable things you can find out through their genetics that you'd be saving the system money.
Look at what we know already. We know so little, but yet we found useful information. How could we not benefit from knowing everything in the genome? It doesn't make any, it seems to make sense to me.
Yeah, it seems to make sense, but there's still a lot of debate about it. There's a lot of debate if clinical grade, 30x whole genome sequencing at $1,000 is worth it to the average patient. And there's a lot of good arguments that maybe it's not worth it to the average patient. There's a lot of even insurance companies.
Yeah, I guess as the price of sequencing comes down, if they can get to the $100 mark, or maybe even to the point where it just, it's like $1, then it would be much easier to get tons of data. But question for you now, so there's various pools of genetic data different organizations or governments have. Do they share it? Can you get in there and look at it? Do they pseudonymize the data and you can look at it, or are they keeping it to themselves and not sharing?
That's a good question. It depends on the data set. It depends how the consents were done. So projects like UK Biobank, right? They have biopharma going in there and mining the data. There was a project not too long ago in Australia where they tried to publicize, just make publicly available so anyone could download it, a bunch of clinical and health record data. They put it online, right? But I think and this is the question you talked about, do they anonymize the data in some capacity? It's so hard to anonymize health data. In this Australia case, for instance, this data set became public, and within like 2 weeks, researchers found out how to trace it back to individuals, and it was a huge fiasco, and they had to take the data set down. But it had already been downloaded thousands of times, right? So anonymizing health data is very difficult, especially if you're talking about genomic data, since your genome is a unique identifier, right?
I was going to say, like, if someone steals your credit card, no big deal, or even your social. If they steal your gene, your sequence, what are you going to do about it?
Yeah,
Screwed.
Yeah, it's true, and it's weird. It's difficult because you can also make the argument that you're shedding your genome all over the place, right? If someone really, really wants to get your DNA, they're going to get it.
Well, sure. That's like saying, the totalitarianism is fine. If you're not doing anything wrong, you have nothing to worry about. It's like the movie Gattaca, where you're in public, so anything about you is we have the rights to. But it's pretty. It's a pretty dystopian view to think that.
Yeah, I agree. But to get back to your question, there are data sets that are fairly accessible. There are varying levels of permissions and access control. Like the UK Biobank I mentioned, I've referenced a few times, is one that researchers are using today.
Well, so I apologize. I've been running you all over the place here. What is the goal of what are you trying to specifically do? What are your goals for this data, and what do you want to do?
Yeah, so we there are a few things behind Nebula. So like I said, our goal is, can we enable medical researchers to really leverage genomic data to its fullest extent? And we want to we spend a lot of time thinking about what are the barriers today, for researchers, what are the barriers for individuals to share their genomic data. And we came to a few of them. The whole sequencing space, especially the direct-to-consumer sequencing space, is backwards, right? You're paying 23andMe and these other companies. You're essentially paying them for the privilege of having them resell your data, right? It's like you were paying Facebook $10 a month, but they're still selling your data to advertisers. People would be angry. That's essentially what's happening today. So the economics are odd, right? It's a lose-lose situation for everybody except the personal genetic company. Users are giving up their data. Users are having to buy these sequencing services. And then on the other end, you have researchers in pharma buying through a middleman, right? They're buying de-identified data. This data has to be HIPAA compliant. There's a lot of ethical concerns. They're typically buying data at a markup, so it's even more expensive. And another huge issue is that it's sparse, right? We came to the issue earlier that genomic data is only as valuable as the context you have around it, right? You need to know the health of the individual, their medical history, et cetera. So with Nebula, we want to build a different platform, a really patient or user-owned direct-to-consumer sequencing company where all data is being controlled. You can get sequencing through us. You can upload genetic data through us. You control your data. You control who has access to it. If someone wants to use it for research, you're getting directly compensated. You don't have to share genomic data. You can share various types of health data. You can get free sequencing if different researchers come and say you qualify for studies. And we have a long-term relationship with our users, right? They're incentivized to come back, to share more information, to learn more about their whole genomes. And that way we can start building these longitudinal holistic data sets over years as we're tracking people, right? No longer are you just going to swipe a credit card, get a box in the mail, spit in it, get your report 2 months later and be done. Instead, we're doing whole genome sequencing. We're really emphasizing that in Nebula. So as new papers come out, new research comes out, we're going to give you more and more reporting, get you to come back and share more information. And all of this is being done in a way that is revolving around user-owned data. There's no backdoor here. We built our platform with a ton of different types of encryption technologies to make sure that your data is super secure. We built our platform in such a way that Nebula doesn't have access to your data without your explicit consent. So for instance, if we get subpoenaed or law enforcement wants to use our database to track someone, we quite literally cannot give them access to the database because there is no backdoor. They have to go through you, through the individual. So we're trying to remove a lot of the barriers that are preventing people from getting whole genome sequencing. One of them is being the cost, because we're letting researchers pay for that instead. They can pay for people to get sequenced. We're removing a lot of the privacy concerns that individuals have when they have control of their data. We're removing the issue of sparse data sets that revolves around genetics, right? Instead of just having a very transactional, swipe a credit card, give someone your genome, answer a few questions, we're building long-term relationships. You can get paid for uploading more data, sharing more information. Researchers can recontact individuals to get them to upload even more information they want. And all these decisions belong to the patient, right? We're not a data broker. We're not selling any of the data.
Well, you did say though that pseudonymizing the data is difficult. So is that a big focus of your attention? The sharing is great, but how do you make sure that researchers get access only to the parts they want, and that the data isn't corruptible?
Yeah, so there's a few things we're working on, and a lot of it revolves around different types of secure compute technologies. So there are ways to do analysis on data without ever exposing the plain text data. So these are technologies like homomorphic encryption, like secure computation. You can employ some of these to allow researchers to run computation on these data sets without ever having to see the data sets. So that's our long-term goal, is enabling these types of technologies for genomic data. And that way we really remove the privacy concerns. There are limitations, right? Homomorphic encryption is slow. You can only do certain types of operations on data sets. Secure computation, there's questions about how secure things like trusted enclaves or SGX really are. People are finding backdoors all the time. So there's a lot of work to be done in the future, but that's where we want to go eventually, is a complete full stack solution for aggregating, collecting health data, and computing on health data that doesn't compromise anybody's privacy, where no one has to see the data. No one can see the data except for the original data owner, who is our user.
Okay. And I guess a question to make it even more difficult, unfortunately, is do you know of anyone that's able to sequence the epigenetic effects on the genome, methylation, et cetera? Can we see that with clinical grade or lab grade sequencing?
So as far as I know, I don't think you can do that with traditional clinical grade whole genome sequencing. There are ways to sequence epigenetics, but it's not something we're doing today. But yeah, I agree. That's something that's super valuable. We want to get into different types of omic data, right? We want to get into panomic. We want to start doing proteomic. We want to start doing microbiome. All of this stuff is useful data that gives us a better understanding of what factors are influencing people's health, and then can eventually lead to better treatments.
Right, well, I don't know if this is a valid theory or not, but I just wonder if epigenetic changes cause changes in the underlying genome. If they do, it's going to make things a lot more complicated. But that's just speculation.
Yeah,
I don't know.
Yeah, I don't know. I'm not an expert on epigenetics at all. From what I've heard that that's not the case. That'd be a good question to ask George next time he's on your podcast.
Yeah, I'll have to have him again. Yeah. So where are you at with your rollout? Are you at the point where you're asking now for people's data, or is this a new initiative? Like, how far along are you?
Yeah, so we launched almost 3 months ago. I think in 2 days it'll be 3 months, on the 15th. We launched November 15th. So we launched our first product, which is a low-pass whole genome sequencing kit that people can just buy. We haven't yet launched a interface for researchers. So this is coming up next. How can researchers compute, run queries on this data? Our first goal is really aggregating enough data that it's actually useful to researchers. So our platform lets people do a few things. One, you can get new sequencing through us. You can just pay for it, and you can get whole genome sequencing. Two, you can upload pre-existing genetic data. So let's say you've gone to another personal genomics company and you've done some form of genetic testing, whether it's SNP chip or whole genome sequencing. You can download that data, which they legally have to let you download, and then you can re-upload it to our platform. Or three, the third option right now is you can just sign up. You can just answer questions, earn rewards for answering questions, health-related questions, and then we'll match you with researchers if you qualify for studies. So we're working with some pharma right now for some essentially pilots for studies they want to recruit for. And if you match with a researcher, you will have the opportunity to get the sequencing for free.
Okay. Well, excellent.
Yeah. So we're rolling out.
What's your goals over the next year or so? Where do you want to get to in terms of number of sequences or number of studies, or what are you looking at doing?
We want to be in the hundreds of thousands of genomes by the end of this year. So that's our goal. And when I say hundreds of thousands of genomes, I mean hundreds of thousands of newly sequenced whole genomes that we're providing through our platform. So our goal is that the 2019 is really for us is a year of growth, right? It's getting to that critical mass where we have enough users in the platform that are voluntarily or choosing to share data, that researchers are finally able to start doing interesting things with the data sets. So that's where we want to get in 2019. We're rolling out a few other products throughout the year that'll hopefully drive more users to us. We have different types of sequencing. Right now it's low pass whole genome sequencing, which it's cheaper. It's around $100, but it's not clinically relevant. So we're going to start launching some clinically relevant products like clinical grade whole genome sequencing and some cheaper versions of that as well throughout the year. So for people who more want to learn more about their health than this infotainment that you get normally from personal genetics, they can do so. We're also working with a lot of nonprofits and rare disease groups. We don't want to get in the trap. A lot of people who have done, like I mentioned earlier, the most valuable data, or the most useful data is data from people who do have rare genetic conditions. So those are the genomes that researchers are really interested in analyzing, and that they can derive a lot of value from. So we're working on projects with a bunch of different rare disease groups and patient advocates to help them build platforms where they can securely and privately share genomic data, which is a huge pain point for a lot of patients in rare disease communities. They're the ones who are their data is the most sensitive, right? They don't want it to get in the wrong hands. They don't want it to end up with an insurer or an employer. but we still want to incentivize them to share the data in a way that's responsible and ethical and helps drive forward medicine and therapeutics for the disease that they're suffering from.
Gotcha. Okay. Well, very good. What's the best way to get in touch with you, Kamal, and to ask questions and suggest collaborations and all that stuff?
Yeah, so people can come to our site, nebula.org. If you want to partner with us, you can reach out to partners@nebula.org. People are also always welcome to reach out directly to me. I'm at Kamal@nebula.org. We're really looking to work with people who can help us drive user growth, right? Help us increase the data that we're generating on the platform. So these are different types of distribution partners. Where we're very interested in working with people who can help us increase the type of analysis or improve the type of analysis and reporting we're doing on the website. So we're launching our reporting in a couple of weeks, which is for all the people who've been sequencing through us so far. They'll now get these big reports, which we hope are very compelling. How can we improve those, generate more valuable insights, especially when we get to clinical grade products? This is all stuff we're interested in talking to people about and working with them on.
Okay. Well, very good. Well, Kamal, thank you for coming on, and I appreciate it. And I hope you end up with tens of millions of genomes that then can be used by researchers, because it's a big deal.
Yeah, I appreciate that. Thanks for having me on.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.